So this is the last session of the day, where we try to use genomics to track the evolution of this heterogeneous organic material. Kind of finding the background where these squiggly lines we have seen in the last session, where we can get them and refer them from. We are in the last module and in the last session of the day. For this session, it's planned that I'm just giving a full walkthrough how you could eventually sample an entire population and extract then relevant information from this, going from the sampling in the greenhouse or in the office, going to the isolation of the DNA or RNA in the lab. Following then, you have to decide how you're going to genotype your DNA. And finally, you have to come up with some idea how you analyze this data, make sense of it. And I want to discuss two different settings. One rather simple one where we know the parents and it's also just a biparental cross, and a more complex setting where we have multiple parents or no reference information of which parent founded this population at all. I want to highlight also some results and assumptions which you could draw by some examples. And at the very end, there is some time if you have questions, and also a small quiz. And this is, I would say, a really small quiz. So no heavy work to do at the end. If you have any questions arising, just send them out as you did during the day. I will try to follow these questions on my second screen. So whenever you have a question, just type it into the chat and I will try to answer it right in the. So we have heard already in the, I think in the first session, how you could generate a composite cross population, and this is kind of analog to organic heterogeneous material. So I brought up here an example where you have three different parents which you cross, bulk them together in a generation of your preference, and then you send it out to the field so that you have then a population that is really heterogeneous and diverse for various traits. As you can see here, colored in here, we have different crossing over events. So this is quite distinct difference from just the mixture of varieties that might also see similarly diverse on the first view, but these here are real combinations by a meiotic event from a crossing. So the question then arises, what do we want to know? We want to know the share of these traits that we find in here, and also how they differentiate over time. And with this, we had a similar problem in the past, a cooperation with the University of Bonn, where they also had a very long ongoing experiment against natural evolution in conventional and in an organic environment, where we also performed kind of a crossing of a former elite variety and some crop right relatives that are commonly used to introduce new alleles that are quite beneficial in terms, for example, for climate change adaptation. And this has been then left out on these fields that you can see here, one organic, one conventional environment for 26 generations without artificial selection. And what turned out to be our question then was, how did they evolve over the time? And this is, of course, also something quite interesting for a breeder who's marketing organic heterogeneous material, as he or her has to present the farmers who are buying this the seeds, how it is changed compared to last year. Who's talking there? I can't hear that, sorry. Right. So a similar question as we had, we wanted to know, and the breeder also wants to know, and ultimately the farmer, what is different to the previous generation and how we can adapt. So this summarizes here these questions. What is happening in these populations? How are they adapting? What impact does the environment have on this population? And with this environment, I'm talking about the climate, the soil, also the farming practice, in terms of a breeder, also the selection, the artificial selection that is performed. So these kind of things are all of interest to us to bring light into the change of the ultimately also the phenotypic appearance and also fitness of a population. So what are we going to do with this? Of course, at the beginning, we have to sample the populations to get an information what is going on. And the very first step would be that we are getting DNA from this population. What we always have to keep in mind, not only in the research area, but especially in the private sector, is that money is a limiting factor. So we don't want to spend a couple of €1,000 to characterize this population, this heterogeneous material. It should be rather cost efficient. So the first step, as I said, getting the DNA, and there are two approaches we managed to establish, which worked quite well. And these are the one is that you directly extract this DNA from the seed. So you grind the seed to flour material and you select a couple of hundred seeds as a representative sample for the entire population. You can think of something like 500, maybe 1,000 individuals, so seeds of a population. And then you grind this to direct flour and then extract directly the DNA from it. What has the advantage that you don't need to grow them in a greenhouse and don't have to spend a lot into labor costs collecting the DNA from the plants that you also have to take care of. On the other hand, it's quite challenging for oil-rich seeds, and also the grinding process is relatively laborious. In the end, the DNA extraction is also a bit more challenging than for typical leaf tissue, but not nothing that can be overcome. The other thing you can do is that you collect fresh leaf tissue. The problem here is that you have to have equally sized leaf discs. So you have to think of that the DNA amount that you want to give into this pooled sample for this 500 or 1,000 genotypes should be ideally identical for each genotype. Of course, this is not feasible to do it in the real-world scenario. Therefore, you try to get to this ideal case as close as possible. And this, for example, you can achieve by cutting out equally sized leaf discs, which is quite labor intensive. With the seed, we usually have equally sized seeds in this population. So by nature, the amount of DNA is almost very similar in all the genotypes, so we don't have this issue. The actual extraction step is rather straightforward, I would say. There are some commercialized sequencing kits that can lysate the DNA and also disperse and all the remaining debris and non-wanted particles of your DNA, like lipids, proteins, whatever, so that you in the end up with pure DNA. This is true for the leaf, but also for the seed. One thing that we have to keep in mind is that we have to grind the seed and also the leaf tissues will affect our DNA fragment size and overall output of the DNA. Because you can imagine this is a beating mill, so we have some metal balls sitting in here that break down the tissue, and with the tissue also the DNA will be damaged and will become shorter. And when you grind this material for, let's say, 10 minutes, then you will just end up with very, very short DNA fragments, which are usually not very helpful. And probably the sequencing company will also reject the sample due to this issue. So the second step afterwards is then that we have to decide on a relevant genotyping approach we want to go on with. We now have the DNA and we have to do something with it, get the numbers out of this. We need to end up with some allele frequencies in the end. And the sequencing approaches, the most relevant I would consider available are the whole genome sequencing, RNA sequencing, so the expression data, so all the proteins that are kind of expressed from the cell will be present in this data set. We are also having a kind of targeted approach to sequence just very specific but also kind of untargeted regions on the genome. This has the advantage that we are contrasting, for example, the whole genome sequencing. We are not sequencing the entire genome, but just maybe like 40,000 areas where we find polymorphisms, like single nucleotide polymorphisms that we can use as a marker. And this usually comes with the advantage that we end up with a higher coverage for these decided regions. Lastly, we have the last generation sequencing, which are Oxford Nanopore or PacBio Hi-Fi reads. These reads have the biggest advantage that they produce very long fragments. So for Oxford Nanopore, it's true that you can achieve as long fragments as you can extract from your cell. So in the past, there have been reported that up to 500 megabase pairs on a single molecule have been sequenced. This comes with different advantages and disadvantages. For example, the accuracy of our data is really high with these Illumina sequencing-based approaches, but our disadvantage kind of is that we have rather short fragments, and all of these three approaches, these fragments range usually from 100 to 500 base pairs. While, as I said, we don't have this limitation in here. Here for Oxford Nanopore, it's kind of infinite. For PacBio, we usually have something like 10 to 25,000 base pairs, but therefore the accuracy is a bit lower of the base precision. As I said, we have some advantages. We have the entire genome. We have quite some high coverage for the RNA-seq, same for the genotyping by sequencing, and this here comes very handy in settings where we have more than two parents present, where we then have to construct an haplotype information. But we come to this point, what does this term mean a bit later. On the other end, we have, as I said, short fragments. I would consider this expression bias even as a bigger disadvantage than the short fragments here for the RNA-seq, because it could be that sometimes, for example, heterozygote species, which is definitely an issue in outcrossing species, that for example, one allele of a gene is expressed but the other is not. Then we have a bias towards the one that is expressed. Similarly, for the genotyping by sequencing, we very often use enzyme-based assays. So these enzymes cut at a very specific position on the genome, and therefore we are not able to identify polymerase chain reaction duplicated reads. So for the Illumina sequencing, we have an amplification step of our DNA prior to the sequencing, and this means that all of our DNA will be multiplied hundreds to thousands of times. This usually does not really matter for scenarios where we check for a single, where we sequence or genotype a single individual. But when we have a pool of different genotypes at once, this becomes a problem. Especially when we are having the same starting and end position, because in these cases we cannot differentiate between an artificial duplicate and one that is already there by nature. So the one thing to keep in mind here is that this here is also quite difficult to use, and therefore it also is ruled similar to the RNA-seq. It has a lowest cost associated with it, but the usefulness overall is quite low. Nanopower packed in a relatively early stage of their development. This is kind of a disadvantage, I would say, but in the end, the usefulness, because these fragments are quite long, is quite big. What we have to keep in mind is that we want to save costs, and cost saving in terms of genotyping and sequencing usually mean that we trim down on the coverage per base. So you must imagine that we are, or the literature advises that when you're doing a pool sequencing approach, you should have something around 1 to 500 fragments covering each base to have relatively secure information about the allele frequency of this specific locus. But I'm going to show you a way how we can work around this and work with much lower sequencing levels. And for this, it's also quite important to know how related the parental lines are. So the less variation is found between the parents, the higher the sequencing depth typically needs to be to account for this. So in the end, once we decided for our sequencing approach, the processing is rather similar. We check for the quality, we align the quality that it's all sufficient to our needs. We use a reference genome of the respective crop where we map all these reads towards. So like a puzzle where we know how the picture should look like. And then all these reads will be mapped to this reference. All the reads that are artificial duplicates will be removed because this will have a quite substantial impact on our allele frequency estimation. Just imagine you have found 10 reads, 5 of allele 1 and 5 of the second allele, but 3 of the 5 reads of allele 2 are just an artifact from our PCR duplication. So instead of having a ratio of 5 to 5, what we observed, the real distribution is rather 5 to 2. So it's quite substantially different. After we did this, we are going to call these genomic markers, these single nucleotide polymorphisms. And once we have that, we can count the reads per SNP that we find. And as these SNPs have 2 alleles, then we can assign these the reads to the respective alleles, and then can make an assumption of the allele frequency for the single SNP. And as we use with a cost-reductive approach, we are going to construct some haplotypes in the end. I will show later on how we are going to do this, but before I want to highlight some technical requirements for this pool sequencing. At least for the easier project, it's quite useful and also mandatory, I would say, to have the parental haplotype, so the parents that founded your population. So we know the alleles of these parents because we have genotyped them, and for the short read sequencing approach, we really need to have this information. Besides that, we need high quality sequencing information. So usually a score from the sequencing is expressed as a Phred score ranging from 0 to 40. And all you have to keep in mind is kind of everything what is green is perfectly fine, and red and yellow are not where we want to be. And ultimately, a reference genome is very, very much useful, and we can get this online. So for the majority of crops, such reference genomes are available. For the most prominent ones, like wheat shown in here, they are also on chromosome scale. And we also find usually some variation database where we find some single nucleotide polymorphisms, for example, that have been reported before. And this is a quite useful information for us because the variant calling in our sequencing data is based on statistics. So the tools that are applied to do this run some statistics on it, and then they will decide, is this base different because this is a sequencing error, or is this a true difference? And most of the times these tools perform quite well, but let's say in 2% of the time they do a mistake. They report a SNP that is technically not present. And when you have millions of SNPs detected across the genome, this means that you have not only two of 100, but you have then maybe 200,000 of something errors in your data, and this is quite a substantial number you want to avoid. So therefore, it's quite useful to use such an approach. So now I want to come back to the point where we want to do this cost efficient. Okay, so there's one question from Linda that I quickly want to answer. What if I want to create an OHM from a crop which has no reference genome? I would say these days it's not super difficult to generate at least a scaffold-based reference genome for yourself, which is also not extraordinary costly. And you can use also these Oxford nanopore reads that we have seen before. So I would say the answer to your question is you are kind of limited to the sequencing approach you want to go for, the long read sequencing, but in the process of defining the allele frequency or detecting the allele frequency, we can use the information of this population you have sequenced to generate at least not super accurate but solid reference genome we could use to align our data against. It's not ideal, but it could be a starting point. So coming back to the presentation, we want to do this for very low costs. Yeah? So already the pooling example, the pooling approach lowers our costs by the amount of how many genotypes we put in the sample, because doing sequencing for 1,000 times for each individual is of course 1,000 times, or let's say 500 times more costly than doing this for a pooled sample. And when we want to even go down much further, that we are saying we are applying the same sequencing depth that is common practice in the industry on a single genotype sequencing of 5 to 10x for each base pair on the genome we want to target. then we are reducing also from this high rate of 100 to 500 reads down to 5 to 10 reads. So that is kind of a 20 times reduction of our costs again. And you can probably imagine that it's quite difficult to correctly estimate the allele frequency from just having 5 reads. So the resolution is pretty bad, I would say, when you are saying, okay, 1 or 4, then you have 20%, and from these 20%, you can actually not be very, very sure that is true. Because imagine there's just coming one additional read to this, and your sequence will not remain the same with this 20% allele frequency for the one allele. It would quite change drastically, so the variance is extraordinarily high. Still, it's possible to do this when we include some bioinformatics and also some biology, including with the reference information. So the first thing that we need to do to estimate this allele frequency at this very low coverage with high accuracy is we have to gain knowledge about these haplotypes. So these blocks across the genome that were not broken up by the crossing overs, and so they remain similar between these SNPs. So our question is which SNPs are linked in these parental lines and which have been transformed into the population via linkage disequilibrium, because these SNPs then remained on the same fragment. And to illustrate this a bit better here, I included this figure here where we see some allele frequencies for 3 different populations. 2 of these populations have undergone already 14 generations of adaptation, and this helps us then to find out some regions that are linked together across the majority of the genotypes. Each of these dots represent kind of a SNP. You can say where we have a relative low confidence, how is the allele frequency really. But when we are aggregating, for example, this big chunk fragment here in the center part together, we can say for this population here, the allele frequency in this chunk part is, 0.3 on average across all of these. Why can we say this? For example, in the end here, it's very clear. You don't see any yellow points popping up here anymore, but they are sitting rather down here. So we must have had some selection events that selected one gene or 2, 3, whatever, sitting on this block here, and then unselecting one or more genes sitting down here in this block. So with this, we can already say, okay, we have quite a number of these blocks across the genome that we can then ultimately use to aggregate this SNP information together into haplotypes to, by mathematical background, increase our security and our confidence in the result and reduce the variance to a minimum. And I want to highlight this here in this graphical summary here once more, because this here is on an entire chromosome scale. And what we generally aim to do is not sitting here on an entire chromosome, but doing it rather for a single gene, or at least a marker we know a phenotype for. Because you see across this region here, there might be a couple of 100 genes that might have caused this increase of the allele frequencies. And our goal as a scientist or breeder might not be to say, okay, it has changed. Of course, that could be an output, but we want to go a bit further with the information for the farmer, that we are saying, okay, this one here has changed because this gene sitting in here is coding for, I don't know, for a longer ear, for example. This one here produces more seeds and therefore has a higher fitness advantage compared to all those genotypes that don't have this entire block. Our goal is not checking this block, but rather going down on gene level. So we are having here our information collected from the sequencing. We have kind of our reference sequence information up here, and these are then our marker positions where we found variations between the parental lines, which we should also find in the population afterwards. When we genotype the population, we find these kind of read depths for each of these loci associated with a given frequency that we say, okay, this allele A is 3 times present, while this allele C is 7 times present. This is not super helpful, but when we annotate this kind of information from these genes to from these SNPs to genes or to markers, we could increase then the knowledge gain. And what we can also do is we can make use of these kind of genes. You can see of SNPs. You see this one here sitting outside the gene, which is very common. The majority of the SMP markers we find on the genome are not inside the gene, but rather in the intergenic regions. When we just extend, for example, our gene here artificially, then we could also do this for a given marker that we know, extend this region here, or even do it for an entire contig, like we have seen before. Then we can aggregate, for example, for this contig, all these SNPs together here, count all the reads here and the respective allele frequencies, and with this achieving a relatively higher read depth, in this case of 85, and getting an average allele frequency score that is not so variant as you are seeing up here from 0.1 to 0.5. And as I said, we want rather do this for the genes. Of course, some of them we can't keep in our data set, like this one here, we just found one gene, but others still have a quite good coverage. Well, how does this now translate into reality? Because this was here kind of an theoretical example that I wanted to highlight. And we also tested this, of course, in a real-world scenario where we tested these 288 genotypes, so 3 times 96 well plate. These were genotyped on different SNPs, once in a pool and once in an individually, and then we compared the allele frequency of the pool to this individual one. This dashed line here would be the ideal fit, so this would be kind of the correct result, and we see quite some difference. As we have heard before, the variance is extraordinarily high. And now when we are using all the SNPs that are close by from the pool sequencing approach and then aggregate this information, incorporate this, and we are very, very close to this line. And as you can see here from this coloration, as more reads we have, as more close we are sitting on this line. So you see this one here is the furthest away. And this one here has about, I would say, 8,000 reads coverage, so really high. And what you can see here from these error bars is that we find 800 SNPs that are associated with this gene here. And the allele frequency for all these SNPs range from technically 0 to 100% for one of these two alleles. And ultimately, in the end, ends up exactly where we want it to have. So the summary is that when we aggregate the reads on the same recombination block, and the gene 99.9% usually always sits on the same recombination block, this can result in a massive cost reduction compared to the necessary sequencing, which is 100 to 500 times X. And this system works very well when you have two parents. And then we can compare these two, alleles to our parental line and say, okay, allele A, that is parent A, and here's the second parent. Very, very easy. And with this, we can also aggregate across multiple SNPs them together so that we are always connecting the alleles of one parent and the other parent by calculating the allele frequencies. So now we are coming back to a more complex example where we have, for instance, here this 4 parents present. We are still having our bulk sent out to the field, and we want to have still the information for each of these genotypes. Okay, I want to know the frequency of this one, this one, this one, this one. So 4 parents in here. So then we have actually quite big problem because what we did before doesn't work out so well. What can we do to overcome this dilemma? The general principle behind this is that we are combining SNPs to haplotypes. And with this, we can differentiate the parents so that you could say, okay, we have 3 SNPs here aggregated to each other, which can be then uniquely identified to be parent 1, similar for second and third parent. And this here is illustrated in this figure here nicely. Our example that I was showing before was more or less we are having this information here sitting in here, and we have also the frequency in which ratio A and G appear. This is not very helpful when we have this 3 or 4 parents in here. We need to have a long fragment that is spanning multiple of these markers, which we then can aggregate to these haplotypes. And then we have this read here, AGT, which we then can associate to one of the parental lines later on. And then we can go ahead again, count how often does this kind of haplotype allele occur in the population, and then assign a frequency to the parent of this population. And the approaches to use in here are the rather PacBio or Oxford Nanopore technologies. So the advantages of this long read sequencing don't stop in here. Of course, we can do this phasing of the haplotypes with a combination of SNPs. But besides that, especially the Oxford Nanopore allows us to identify this methylation information. So the epigenetic variation in the populations can also be assessed from this. And once we know the haplotype from the SNP combination of a read, we also know that this given read is associated with parent 1, 2, or 3, and then we can associate this methylation information to this parent as well. So we can also do a kind of methylation frequency comparison. And lastly, what we can also do, or as an advantage, is that we can check for the microbial composition of a given population based on these long fragments. This is the fact because in assigning a given sequence to a microbe, identifying which fungi or bacteria or whatever is it, you compare this to a big data bank, and usually this is done by small areas on the genome, which you can hardly target by this Illumina short read sequencing because they are too big. And with this one here, we have on the one hand side the possibility, as the reads are longer, to span across this entire region, but we also have the possibility to not only select these regions to compare, but we can use technically the entire genome of a microbe to scan for its DNA and then compare it to a database. And this can then be quite useful to distinguish also between short-term adaptations characterized here in the methylation pattern, and also specify, for example, the environment, identify the environment these populations have been grown in. For example, you could think of that you're finding microbes that are very special to conventional farming in a batch of seeds, and for another batch, you find some microbes that are rather more likely to be found in organic farming. So now we are going one step further and want to know how this phasing of these haplotypes might look like in the background of these long read sequencing, where we have to concatenate multiple SNPs on one of these sequenced DNA fragments. I'm just giving you here a short overview of how things are managed and done. Run our sequencing as before, then we perform some in the background of the Oxford nanopore. We have to identify the correct base pairs from the raw data that is created. We then again clean our data and align it to the reference genome. Afterwards, we perform again this variant detection, where we find all the single nucleotide variants across the genome. And once again, we are only extracting the polymorphic loci. We detect in case we have a parental, we have the information of, for example, the 4 parents going into this population. And once we have this kind of information, we use this here as a kind of additional reference next to the reference genome to phase the single reads we find. So we still have millions of reads that we have to process, where we always try to find this combination of SNPs across the reads, and then we, for example, drop reads that have a poor quality, that are not long enough, or that have less than 3 SNPs. And after this, we are building then from these single reads some haplotypes across, for example, contigs or genes. In this particular case, we have really lowered the sequencing to minimum approach possible. So we decided we are looking into contig windows. And how does this actually look like? This is quite abstract, shown in here. So we have our reference, and we found these SNPs across this. We know here are these SNPs sitting, so we also have our aligned reads, and then we are picking each of these reads that we find in here. And we are checking on each of these positions. Okay, please give me the information, what base is present at this position. And we have these 6 SNPs here across this read, which gives us then this haplotype. And the real world data is looking something like this. We have here our position where we are mapped with the starting position on this given chromosome. We now know how many SNPs are present, and then in the end we know this haplotype string. And this haplotype we can then assign to one of these 4 parents that we know to perform later on the calculation of the allele or haplotype allele frequencies. Besides that, I want to highlight how it's done during this methylation. So we are again using the Oxford nanopore reads. This is kind of a unique feature for this, that we also can estimate the epigenetic variation from these reads. Everything remains similar until the alignment, but afterwards we are running some methylation detection, and we are also extracting these patterns for regions which are usually annotated also to a gene. The majority of the methylation is happening either in front or in the back of a gene, and these regions we are going to target, and then you can compare environments or generations specific to these methylation patterns. And lastly, what I want to highlight is a haplotyping pipeline where we don't know the parents. This goes into the same direction as it has been asked before. When you don't have reference genome, we don't have now these parents as a reference node. So this picture where I've shown to you, we have this ATG, CTG, and so ever extracted, and we can directly annotate it to one of these 4 parents that were used to establish this population. This is no longer possible. And in this particular case, we have to walk around it, that we construct this reference of these SNPs ourselves. And this is, made by constructing quite long contigs based on the SNP profile of these reads. And we can also aggregate these contigs then later on by matching the overlap of these reads. And usually you make use of principal component or k-means clustering of these specific reads, that you try to get a probability of reads that are more likely to be collinear. And once you did this, you just proceed with what you have done before, that you phase your single reads according to your reference, and then you run this haplotyping step. So now you might wonder, okay, what information can we actually gain here? It kind of depends, I would say, on how much you want to spend in terms of money. So the first question is, are these populations different? Also on a statistically significant level. And you can achieve this by very low cost with an ultra low, also short read sequencing approach. On a second level, you might wonder, okay, what has actually changed? So which regions are different? We have seen this picture of the chromosome scale linkage blocks. So this is something we would be able to see with this low sequencing approach. And ultimately, we kind of want to see gene level differences, maybe also methylation patterns and microbial variations, and therefore we kind of need a medium-low sequencing approach where we then ultimately can pin down genes that were causal for, let's say, most likely genes that were causal for this change. So with this, I want to give some examples here. So the lowest level, what are the populations different? You can actually see on such a principal component analysis plot where we, for example, have a population that's coming from a breeder and then sent out to 2 different farms. And we can then over time see that these 2 farms evolved quite differently and have adapted to their local environment. What we could also see in this example here, where we tested more generations, we see quite linear change in this example here until here, and then we see a big selection force coming in here that is changing quite dramatically the population in here, but on the other farm there was no such a big difference. And what we can also quite well identify is, for example, this one here. You might see here in the back there is some brownish area that should be the second replicate to this one. So we can quite clearly see which sample have been labeled in the wrong way. Yeah, besides that, of course, we want to go on the second level, and with this here I brought an example of spring barley, where we see here the allele frequency of the third generation before it was sent out to do different farms. We also see some very prominent genes here and their position marked in here. As an additional information to this population, this has been backcrossed 2 times, so this black line kind of indicates the allele frequency we would expect in an infinitely big population. For example, on the first chromosome, we see that this line here is matched quite well, but for instance, on this 5th chromosome, it's not matched at all very well. So we have almost 0% allele frequency of in this case a wild crop-right relative that was used as one parent, as one of two parents in this population. So let's have a look here on this fifth chromosome, and we will see how it has evolved over the generations, because now we are in the 12th generation, so 9 years after. I'm jumping just once back so that you can see this once again. This gray block here in the back, this is a centromere, so we usually see little recombination happening in here. And once again, we jump forward and we see not much happened up in here, but here we see, okay, we are from 0. This red line here is what we were starting from, to almost 40% in here. And then we are sharply going back down here, again to 0%. Okay, let's go a bit further. 16th generation, 22nd generation, 23rd generation. We still see this quite big peak. We see here in the gray line representing one farm that this entire block here has remained quite stable. But for the other farm, we see a bit more kind of selection pressure, where it's also differentiating in between these different loci here as well. So while the fitness effect in the gray farm, the gray population was neutral for all this area here in the yellow, for the yellow population. On the other farm, it was actually not, and you could say, okay, this here was rather beneficial, but we see here is clearly some negative selection for this, and this spots in here. So let's go back and check out another region. For example, this chromosome 3 in here. You see, it's below the line also, but not much. We are going onward and we are seeing here some changes going on over the generations as well. And we again see there's not much fitness benefit from the crop-right relative for the population, for this gray population, but for the other there is. Of course, there are some regions where there's no benefit at all. For example, this one here, this one, this one. But for the ones that increased in their frequency, there actually is. We are coming to this plot shortly back when we are going on the last level, on the highest level we want to check on. Because shortly I want to highlight here also how it would look like if we are checking for more complex population. For example, when we have more than two parents in there. And there we kind of see a similar pattern. Yeah, we use this Oxford nanopore sequencing I described before to differentiate then these populations. And for instance, we would see this big block here for this genotype here got up selected, also very sharp edges here, so there must be a crossing over event for not only one genotype, I suspect, but we see something similar also in here and here. And you can see that these blocks here become smaller and smaller as further away we go here into the telomeric region away from the centromere. This is just true because we have more recombination events in this region compared to this region here. So now we are coming back to the figure we have seen before, and I want to highlight with this the highest level. Identify candidate genes which are causing the selection or are candidates for selection. I highlighted here these 6 different groups, arbitrarily chosen. For example, biotic resistance or resistance genes, yield, physiology, for example, the yield, not the yield, the flowering time, how they perform root rise, the yield components like how many kernels per seed, the total yield, drought tolerance, and also where they are in, for example, nutrient use efficiency. And all these here got some little dots in here because people in the past have done some genome-wide association studies when you have taken part in the class on Tuesday, you might know what this means, and they identified some regions that correspond to a specific phenotype. And for instance, this one here, highlighted here, where is it? This one here as yield component. It's not very accurately assigned in here in the figure, I guess. No, sorry, here, yield physiology, sitting somewhere right here. We know that there is one gene that is related to brittleness of the ears, so how easily it falls apart. And in a system where we have a crop rotation and the plants move from one plot to the other in the next year, it's quite yeah, I would say a useless trait that your ear is so brittle that it can't be harvested anymore in the right time by the combiner and then sawn out next year. So this is something that would be unselected just because it has a low fitness in the given farming environment. And analog, we see something similar down in here, where we have 3 dots sitting in here. One is a yield component and the other one is yield physiology related. One gene that was identified sitting in here is a dormancy gene. So a gene that prevents the seed to emerge, even for multiple years. Similar effect, even stronger than with the brittleness. We have a negative fitness effect in a scenario where we have a crop rotation. And on the other hand, we are having sitting up here on top very closely a gene that is related to the ear size. So we have more kernels with genotypes having this specific allele, and therefore the fitness in such a population is quite high. So what we want to do next is kind of extract the frequencies across these 3 groups. This is just an example what you can do, and then check across these generations how the frequency of this crop right relative has emerged. And for instance, we could see that the roots develop quite differently, in this case between an organic and a conventional farm, but also the drought tolerance and the yield physiology are quite a bit different. So our next step in also developing this method was we wanted to validate this information. So we are going into this aspect here, and yeah, we already have converted this frequency information into this phenotypic characteristics that we can say a farmer or the breeder as a general information. Okay, we see more wild alleles present in one population than the other for root-related phenotypes. And we, in one publication, we also checked on this, how it's looking really like for the root length and also the root mass density, how many root tips we have, and the root length. And we could see that, especially I highlight this now here for the root length, that the difference here is statistically significant. And what you can also see in this particular example is that the diversity of the genotypes in here, so the span going from up here to down here, is much higher in this organic coiled population than in the conventional coiled population. And this is more or less true for all these 4 genotypes. And yeah, I want to stress out this is an experiment we conducted after we did this genotyping of the population, just to make sure that we are not finding something that is not correct. So now you might wonder, okay, allele frequency, very nice. We have heard so much about this today. Can't we go back to more true scenarios where I get just a simple probability score, a p-value, which is indicating to me, okay, this region here has been affected by, for example, adaptation by selection. Yes, there is. There's one very nice tool published by Mathieu Gauthier that can extract a p like value. It's not actually a p-value, but it behaves similarly, so that you can say as higher this XTX score becomes, as more securely you can say this one here is associated to an adaptation process. And when we apply this to a population that we were working on, you can see the chromosomes are now a bit different. So this is the rapeseed population. We can see that, for instance, here this organic farm, the population on the organic farm has no big peaks shown in here, but the population on the conventional farm indeed has quite some peaks going up here. And these peaks here are related to the drought response of this population. So what does this mean as an interpreting from a science perspective? This could mean that this population was already well adapted toward a drought response prior the drought events happened, and therefore didn't face any drought-related issues. So it didn't need to close the stomatas or whatever. So the set of individuals, the genotype frequency of this population was well prepared for drought events. While for the population adapted in this conventional environment, this might not have been the case, and this change in the climate scenario also corresponded to a effect. So the allele frequencies were changing toward genotypes with a higher drought resistance. So besides that, I also want to highlight this fact about this methylation profile, because this is also part of the genotype, and we can also extract some probability scores from this methylation profile when, for instance, we are comparing two different populations, as these population organic versus conventionally managed. We see some regions in here, and when we are digging into, for example, this one here, we can, for example, identify how many of the CPG islands, or how they are called, are methylated or not methylated per this respective environment. And as more these are methylated, as lower the chance are that this gene is actually expressed. And these can be quite stable, but also can be changed across generations, also faster than the allele frequency. And you could potentially also have a variation in this methylation pattern without having a variation in the allele frequency patterns. So the allele frequency between two populations can be exactly the same, but the methylation might be quite dramatically different. And this means for this gene, which is a polynucleotide transferase, this is telling you maybe even more than it does me. But when we are going for literature research, we find some autolog genes, in Arabidopsis, for example, which showcase that these peptides, such proteins, are relevant to mediate environmental stress or plant defense response. So you could imagine, okay, this gene here is quite relevant to be active in resistance breeding, for example. So just to highlight that there is also another level than just going on the plain marker level of the genome with the single nucleotide polymorphisms, but going on another scale. And similarly, you can do also the same for the microbiome. So that you, for example, could compare the seed microbiome of a given population and compare it across all these generations, where you can, for instance, I picked this example here of a Stenotrophomas genus, could say, okay, we see some variation in the frequency. We find the DNA associated with these with this genus, and we know from the literature also that this one here has a positive effect on the germination rate, for example. With this, we could, as a breeder, highlight to the farmer, okay, we have some material that is coming from this particular year, which is relatively high in this bacteria that is promoting the plant emergence rate. So acting similar to a seed treatment in a conventional environment, for example. So I want to highlight here a short summary, what we have seen so far. So we can genotype an entire population and extract a lot of information for little cost from this. We can see also changes across generation, and depending on how deep we sequence, we can track this with a very high precision down to genes, and with this extract then some phenotypic variations and predict this. And the advances in the latest sequencing techniques also allow that we can nowadays track the epigenetic and microbial variations in this organic heterogeneous material. Yeah, so far, so there's a bit more to it I want to highlight. Not much, but more. So we have already said that there is something more we can get, and this could actually be the recombination rate we could detect. I have explained before that the allele frequency changes over time kind of uncover all the recombination events that are present in these populations. And for instance, we counted here roughly the recombination events for each kind of chromosome. And here's a kind of summary for these two populations. We compared the conventional to the organic one, where we can see that the organic has more recombinations in there after 20 years of recombination. Does this mean that it gathered more recombination over the time of adaptation? In this particular case, the answer is definitely no, because this is a selfing crop, self-pollinating crop. So I would say 99% of all recombinations that are in this population were coming from these initial crosses, and then they were muted. We couldn't see them. For example, like in here, we couldn't see them because the allele frequency was not. But with some adaptation due to fitness events, we can see that there are some changes becoming visible that we could say, for example, here, clear a crossing over happening here as well. Here, there might be some more. And we have seen this example before that we are having here much more crossing overs going on in this area in here. So we can also harvest this kind of information from this population data. And why is this kind of interesting? I mean, as a farmer, I would say I wouldn't care much, at least me personally. But for a breeder, this might be quite interesting because this can be also spawned to not only different environments, but also to different parental lines we use, because each genotype has an individual crossing ability and also an individual recombination rate that is associated with this. And when you are using genotypes that are more prone for recombinations, then you can see this from such pooled data. And you technically don't need to wait for 24 generations, but you can already see this in the third to fifth generation with a little less precision, but still you can see this. Ultimately, you can also identify the recombinations coming from this long read sequencing, because kind of every sequence, every of this haplotype you can't associate it to one of these four parents, means that there is a recombination break event happening in here. And then you can identify regions inside these blocks that are more prone to have a recombination happening in there, so-called recombination hotspots. Yeah, with this, I want to give some final conclusion. So I hope this was quite useful and helpful to give an overview how you could detect how this organic heterogeneous material changes according to a different environment or years. And one additional thing, it could also serve maybe as an additional support. Maybe some of you know that there is an OHM track system in development where farmers can then, breeders can show a farmer how a population seed batch has developed in the past, so that the farmer is not buying something he can't really trust in. But yeah, when you want, or if you want, you could also pin this down a bit more precisely with this genomic approach, that you might in the end up with such a card that is referring to your population you have purchased as a farmer, having certain attributes. And after 3 or 6 generations, you can then provide them with a given information about these populations. For instance, that you're saying, okay, the population has gained quite good power in rooting, but the yield potential declined over the period quite dramatically. And yeah, it might be time to re-establish this population with new fresh seed material. I think this question was also asked in the first module today. Yeah, with this I want to highlight for those of you who want to receive the certificate for today's entire module to yeah, finish this quiz. Either you go by this link, which I will share in the chat shortly, or you scan the QR code, and please do this by the 17th of March. This would be quite helpful for all of us. Yeah, with this, we are at the end of this presentation, I would say quite well in time. If anyone has an additional question to the one Linda asked, you are free to ask.